When we sat down as a DDF group to say, what are we going to invest in? We talked about increasing the scope of approaches, and we looked at the scientific landscape. And the first thing that we realized was that there were major advances in understanding cell biology and its pathology in other therapeutic areas, in oncology, in immunology, in metabolic medicine. And in a way, we can hypothesize that what's happening in the brains of people with dementia is that their cells, the neurons and the glial cells that support them, are undergoing, in some cases, very long-term, lifelong dysfunction. And so things like mitochondrial function, where the mitochondria serve as sort of the power plant of the cell, but also have other important functions in regulating normal cell behavior, these things can become abnormal with age, and that we can develop new treatments that target mitochondria and how to normalize them. Another example is microglia. The microglia are the native immune cells of the brain, but they have really varied function. They eat amyloid plaques, they eat dead cells, but they also seem to be involved in a way we don't fully understand in keeping synaptic connections healthy. So targeting those microglia, returning them to a state of normality, is likely to have real benefit for people in a range of diseases. Synaptic physiology itself is something that we would like to target. It's a big, broad area, and we're talking to leading academic experts in other fields of neuroscience as well as in dementia to understand what we can learn about these things. There's a final area where we're focusing our investment strategy. It started with the observation that ApoE4 is the best known and most common genetic risk factor for Alzheimer's disease. We know structurally that it differs from ApoE3, but we know very little biologically or physiologically about what makes it different. And when we started looking at how to explain differences between ApoE3 and 4, we realized there's a very rich biology that comes at places where membranes from organelles are in contact with one another, called membrane contact site biology. So we've started, within DDF, we've started 6 projects from either very early companies or companies that we've created, each of which are investing in some of these aspects. We have a membrane contact site project. We have a company actually that we formed called Tiaki Therapeutics, very early seed company which focuses on microglia and how to normalize microglia. We've also invested in 3 companies, Elector, which is using immunological approaches to try to treat a range of dementias, Mitoconics, which is a mitochondrial dysfunction company, and a company called Ceravance that has a unique platform for helping us understand what's going wrong in specific cell types in the brains in dementia and how we might find new protein targets that way. So there's a lot out there. There are many places that we can intervene. And what we think is that dementias, Alzheimer's and other dementias, can start in any of these places. It might start with inflammation. It might start with mitochondrial dysfunction. And that sets in motion a cycle, not necessarily a cascade, but a cycle in which each of these dysfunction leads to further dysfunction. So mitochondrial dysfunction damages cells in a way that activates microglia, and microglia activation alters the metabolism of amyloid or tau. So all of these are places where we can, and we think we should intervene.